We are picking up Chapter 6, Part 2, with the storage of specimens. Urine, viral blood specimens, catheters, and swabs can be refrigerated. Also, respiratory and stool cultures can be refrigerated, but it's not best. We usually want to do those pretty quickly. What needs to be processed as soon as possible or immediately, blood cultures and spinal fluid. So blood cultures will come in bottles with a barcode and patient labels, and those are going to go into the blood culture machine, and it's a machine that rotates the blood culture bottles constantly and monitors any change in atmosphere within the blood culture bottle. So it will flag it as positive. So those need to go in the incubator as soon as possible so we can get those going. And then spinal fluid needs to be centrifuged to concentrate any potential bacterial pathogens that may be in there, or any pathogens if we're doing fungal cultures or mycobacteria cultures. We have to centrifuge that, because if there's just a few organisms, we want to increase our chance of isolating those. So we're going to centrifuge the spinal fluid, then take a sterile pipette, and we may not be able to see any button or concentrated stuff in the bottom of our sample, but just take your pipette all the way to the bottom and aspirate the sample, and then put that onto your direct smear. Put a drop onto the gram stain slide and the media. Things that can be stored at room temperature would be specimens for fungus cultures, anaerobes, genital cultures, ear, eye. Those can stay at room temperature because they're usually going to be on a swab. And then, we talked about other things that can be refrigerated. All right, so we're going to move on to the packaging. We talked about this in a previous chapter, but this is a picture of the mailer that you would need to use. There's going to be a box on the outside, and then there's a tube with a tube within a tube within a tube. So you have the outside box, the inner secondary packaging. Usually this is a cardboard tube with a screw cap. Then there's another cardboard tube with a screw cap that fits inside. And then there will be like a biohazardous bag with some absorbent material in the middle. And then your primary receptacle will be in the middle. But you have to have that absorbent packing material. And it's kind of like the inside of a baby's diaper. If it gets wet, it absorbs like a whole bunch of times more than its original volume or weight. But this is what you'll have to pack the samples in to go out to the health department. And there's a whole 8-hour class that's taught by the health department every year. So if you work in the shipping department where you have to do send outs or send things to the health department, somebody in the department has to go to that and get certified to package infectious substances. It's very involved. Unfortunately, we also deal with unacceptable specimens, and we have to reject these specimens. And this is a big source of tension for us, because people don't like it when we call and have to reject their samples. But remember, the main goal of specimen collection is to maintain viability, decrease contamination, and we want to report accurate results. And if we don't have an appropriate sample, we cannot do that. So, some unacceptable samples would be if there's no label. If a label and a paper requisition on the specimen do not match, we can't really verify that that sample does belong with that requisition. And also, we need to consider how invasive was the procedure before we just reject something. For example, if there's a piece of brain tissue in a cup that's not labeled, chances are there's only one patient that's had some brain tissue submitted for culture that day, and you can figure that out. But technically, we are not supposed to accept any unlabeled specimen. If it's been transported inappropriately, if it's leaky, if there's a syringe, if it's been too long for us to culture, if the specimen is already in fixative. So when this happens, usually it's that cytology or anatomic pathology is bringing us a sample and saying, hey, this tissue needed a culture, but it's all in this fixative. Can you use it? No, we cannot. So we would have to reject the sample. All right. Here are the levels of priority. So when you're slammed, when you have so many cultures that you don't know where to start, you have to know which ones take priority. So level 1, the ones that you want to do first are either critical samples or deeply invasive. For example, spinal fluid collection is really invasive. Heart valves, pericardial fluid, brain, amniotic fluid, those are considered precious samples because they are hard to obtain, not easy to recollect, and so we want to get those done first. Level 2, these are the unpreserved samples, body fluids, sputum, bone, stool, drainage from wounds, tissue, anything unpreserved, that's going to go next. Then the third level is anything that requires quantitation. For example, urine culture, cath tip, tissue for quantitation, and it could be a bronchial lavage for quantitation. Anytime you have to do a colony count, that's going to be number 3, because the longer the specimen sits, the more, the greater the chance that bacteria can multiply and falsely increase your colony count. Then number 4, level 4, the ones that can sit until the end of your shift, because we're always going to clean up our lab bench, right, and make sure that all of our specimens are processed before we go home. Those would be stool and preservative, urine and preservative, and any swabs in holding media, both aerobic and anaerobic. Okay, back to specimen rejection. We covered those pretty well. I want to bring your attention to the last one on this slide, expectorated sputum. If there are less than 25 white blood cells and greater than 10 greater than 10 epithelial cells per low power field and a lot of normal flora, we are going to reject that. But we are going to talk more in depth about how we evaluate sputum. Basically, sputum, you need more white cells than epithelial cells and not a whole bunch of mixed bacteria. Before you totally reject a sample or throw it away, you need to call the physician, make the call, or the nurse, or whoever collected it, the clinic, and let them know that you need a new sample. And a lot of times what we'll do is we'll leave that specimen, and we'll put a sticky note or post-it note on it that says rejected, called, nurse Susan, the date, the time. We'll send recollect, and I hold it right there until you get the specimen that is correct. That way you won't forget about it. And make a note. Always get in a habit of always, especially even with the college, with businesses, anytime you ever have to call anybody for a problem or to get something done, you need to take down the person's name that you talk to so that when you call back or you show up for whatever it is you wanted, and they'll go, I don't know what you're talking about. Who did you talk to? And it's always helpful if you know who you spoke to and when. Okay, we also have to do a macroscopic observation or gross exam. That just means we look at it with our eyeball. And if you work the receiving bench, you'll be responsible for recording the total volume of a specimen. For example, spinal fluid, if you get 4 tubes of spinal fluid and there are 3 tubes that have 1 milliliter each and 1 tube that has 1.5 milliliters, you would report that we received 4.5 milliliters of spinal fluid. And usually that goes to the chemistry department and they put that in for everybody. But you'll be the only one that has all 4 tubes in your hand at one time. So you will probably be responsible for recording that. We are going to note the presence of blood or mucus and look at the general appearance, especially if it's a fluid. Remember back to that spinal fluid, you're going to need to note if it's clear or cloudy or the color. Is it pink? Is it yellow? Xanthochromic? And all these things, the macroscopic observation helps determine the adequacy of the sample and if there need to be any special test. Microscopic observation, this is the direct exam. And what we're doing with the direct exam is we are trying to determine the quality of the specimen. And the quality of the specimen is going to help diagnose the disease. It's going to guide the routine culture and interpretation. It's going to dictate the need for non-routine processing, like do we need to add some special media? Do we need to add a special latex agglutination test? And this will also guide the antibiotic therapy. Now, we don't tell the physician if there's a whole bunch of normal flora on the gram stain. He doesn't need to know that they're gram-positive cocci in change, in clusters, gram-negative rods, gram-positive rods. That's going to be a bad sample. But you don't want to over-report the bacteria that you see on direct exams, especially from a sputum, because it could just be normal flora. And that confuses people. The end culture result is there was only routine normal flora present, no pathogens found, no pathogens isolated, but you had all this bacteria in the direct smear. That's confusing. Also, you can get rapid information. Some of our direct smears can be diagnostic. For example, if you have an India ink and you see a yeast with a halo, that's going to mean it is Cryptococcus. And then if you have a direct smear from a genital sample for a male, if there are, for example, intra and extracellular gram-negative diplococci, that is going to be diagnostic for gonorrhea. Okay, we are also going to be responsible for selecting media for these different sites. So we talked about media selection and the different types of media in Chapter 1 at the beginning of this Pathogenic Micro 2 course. So the different media is going to be specific or geared toward trying to pick up certain types of bacteria. For example, we talked about how Mahon Ch is a differential agar, and it is going to grow gram-negative rods. And then there's another agar called CNA, or for Columbian nalidixic acid, and that is going to be for gram-positive organisms. There's enriched media like chocolate that we talked about, blood agar. There's also enriched broths that we're going to talk about. For example, Lem broth is specific for group B strep. It will keep normal flora growth inhibited while it allows group B strep to grow. And then there's just a broth media. We're going to use some in our class. It's called thioglycolate broth, and that grows pretty much everything, even anaerobes and microaerophiles. So here is, this is going to be helpful. Table 6.4, you can refer back to this all semester and even during clinicals. This tells you the specimen. So first, let's look at spinal fluid. It tells us what test do we do a gram stain and what media or broth are we going to use to set up for spinal fluid. We're going to do a gram stain. We're always going to set up a blood agar plate. We're going to set up chocolate. We're not going to do a Mahon Ch because we're not going to expect a lot of gram-negative rods. But if we do, it's going to grow on your blood agar plate, and you are not going to have a lot of bacteria in your spinal fluid anyway. So likely there will just be one organism, and a blood agar plate is fine. Your chocolate will be for Haemophilus or Neisseria organisms. And then you're going to do a thio. So if it happens to be an anaerobe, notice we don't go ahead and set up an anaerobe plate. This thio broth will pick up any anaerobes. And look, other tips, centrifuge recommended for gram stain. So this table will be helpful. I suggest that you keep a copy of this, save it to your computer, and this is going to be a great reference for you to come back to, table 6.4. And it's quite long. It has all of our samples, stool, GI tract, genital screens, tissue. It's going to be very helpful for you. For specimen preparation for direct inoculation, that means what samples are likely going to get a gram stain. Pus, we are we're not going to do urine gram stains, but sputum, sterile body fluids, and the concentration to improve yield, we are going to centrifuge fluids greater than 1 ml. I've never centrifuged anything in the micro lab at 20 minutes. For 20 minutes, it's usually 10 minutes, and that's good. And there's the image of the centrifuge to help you remember that we are going to centrifuge our fluids that are greater than 1 ml. Also, swabs. You will probably be getting 2 swabs sometimes and 1 swab other times. The best situation for swabs is to get 2 at one time. 1 will be for a gram stain and 1 for culture, because once you use a swab to make a gram stain, it is no longer sterile. So if you should only get 1 swab, you want to inoculate each type of agar that you need first, and then as the last thing that you inoculate is your gram stain. You want to spread that out on the slide for the gram stain. And just a heads up, you may get some tissues in the micro lab, like a toe or a leg biopsy, and you might need to cut off a small piece, and you're going to add it to a small tube. This homogenation system kind of works like a mortar and pestle. So you would put your piece of tissue in the bottom of this homogenizer apparatus, pour a little of your sterile thioglycolate broth in the bottom, and then take this plunger. It has kind of grooves and teeth on the bottom, and you're going to twist and squish. And then at the end, you would use the homogenate liquid to do gram stains and set up all your cultures. Now, let's go back and remember this. Direct smears are not useful for throats, nasopharyngeal swabs, urine, the female genital tract, or stools. That is line 89 through 93 on your study guide. So again, direct smears are not useful for throats or nasopharyngeal swabs. There is too much normal flora. Urine, that is not helpful. The female genital tract, there's also too much normal flora for that to be helpful. However, in men, the gram stain or direct smear can be diagnostic for Neisseria gonorrhea. So it is helpful for men, but not helpful for women. So remember that. You may see it on a test. And also, stools, we generally do not do gram stains on stools. It's not helpful for any diagnostic purposes. However, you may do a gram stain stool for something else, and we'll talk about that later on when we talk about GI pathogens. Okay, so in lab, in the first week, we are doing isolation techniques, streaking our plates, and we are learning about 2 different methods. There's the isolation streak, where we have 4 quadrants when we're done spreading our sample onto our plates, and we have the quantitative isolation, where we are doing a colony count. So here is our streak plate. In the first one, you start out with some sample. In the first quadrant, you're either going to put, if it's liquid, if it's a fluid, you're going to put a drop right here, and then you're going to take your sterile loop and smear it out in the first quadrant. And we're going to practice this with pencil and paper before we actually do this in the lab. And there's a technique to it. It's kind of like trying not to put marks in Jell-O because you don't want to tear up your agar. That makes it dry out more. So we're going to practice. So your sample would go here, and if it's a swab, you would just roll the swab back and forth in this first area. Then you would take your sterile loop and then try to drag it out in the first quadrant. Now, I don't really like this image because they don't go into their first quadrant at all, but we would then take our loop and start over here and go into our first quadrant a couple of times and then drag it out. So we would end up, this would be our second quadrant. Then we would take our loop, and they've done it here. This looks pretty. They take their loop in a couple of times and then drag it out. So this is the third quadrant. And the fourth quadrant, you would drag your loop into the third quadrant and then smear it out. So the goal is to get isolated bacteria out here in the fourth quadrant. So We have the first quadrant that will be most concentrated because this is where our sample went first. And then in the fourth quadrant, this is where we will have the most isolation. So please make a note of this. This is very important, and you will be using this throughout the whole course and the labs that we do, and in practice when you get to the microbiology lab. This would be something that they expect you to know. Now, the colony count, we use a 1 microliter loop. It's a loop with a small hole in the end that holds 1 microliter of liquid or fluid, urine, whatever. Usually we're doing colony counts on urine. So we would take that 1 microliter loop, draw 1 line down the middle with our sample, and then take the same loop and then try to smear it all over the plate. Just take our pen and do that. And then we would incubate that for 18 to 24 hours, and then the next morning we would count the colonies that are growing. And so if there were 3 colonies growing the next morning, that would mean there were 3,000 colonies per milliliter of urine. And don't worry, we're going to go into that a whole lot more. So you also need to know that most cultures grow between 35 degrees Celsius and 37 degrees Celsius, which is, of course, body temperature. We are going to learn how to decide whether we incubate our cultures with a little extra CO2 or without oxygen. And most routine cultures are held from 48 to 72 hours. If we need to hold cultures for 5 to 7 days, whoever collected or the nurse physician is going to need to let us know, hey, we're looking for this odd bacteria. Please hold this culture for 5 to 7 days. Okay, and we're going to talk about these earlier. These are the unusual or fastidious bacteria, and things that fall in this group would be like Bacillus anthracis. That's the bacteria that causes anthrax. We would need to look for a few different things and hold them for at least 3 days, etc. But we're going to cover all these later on in the course. So the specimen collection chapter has been about preserving pathogens and keeping normal flora contamination at a minimum, because that is going to slow us down. So when we do if we have a potential pathogen in the micro lab, we have to quote unquote work it up. So before we decide if we're going to work up a bacteria or if it's normal flora, we have to consider the specimen source. Does that source have normal flora, like a respiratory sample, a throat sample, a sputum? And also, what are the most likely pathogens? What is that pathogen going to look like on these different agar plates that we're using? What is the colony color? What is the shape? How fast does it grow? Is it big? Is it small? And also then, which medias demonstrate growth? Is it growing on McConkey? That would mean it was a gram negative. Is it growing on CNA? That would mean it's probably a gram positive. And does it require a full workup to the genus and species? Not always. Okay, non-routine specimens. This would mean if the specimen is likely to contain low or high numbers of organisms. We might need to use a broth. So in a lot of our cultures, like wound cultures especially, we're going to also set up our broth cultures, and that will be held for 7 days. So we're going to check that to see if there's any growth for 7 days before we completely finalize that culture. Okay, so I love this because this is what we would see typically from a respiratory culture. And what's your thought when you see this? So first I want to point out the quadrants. So this was likely a sputum sample or a throat swab. So we would inoculate the first quadrant, like rub our swab or put some sputum there. And then we would switch to a loop and we would spread this out across the first quadrant. Then I can tell the second quadrant because the lines start going a different direction. So they went into the first quadrant a lot. Okay, so you can see very concentrated, then slightly less concentrated, and then we can see the third quadrant being pulled out. into the bacteria being pulled out to the third quadrant, and then finally the fourth quadrant. So when you first see this, you might first think, oh my goodness, I'm afraid of this. I don't even know where to start. But then we start to ask the questions. What sample is this? What pathogens I'm expecting? We're going to also check our other plates. Is there growth on McConkey? Is there growth on the CNA? And then we're going to look for isolated colonies. So whoever did this streak plate, this looks really good, because this is our goal right here, ladies and gentlemen, before we can work up any culture, which means identify it, the bacteria, to genus and species, and tell the physician which antibiotics can be used. We have to have isolated, pure colonies to do our testing in microbiology. So this is really a lot of fun. Okay, so another positive for working in the micro lab, it's usually you're by yourself, there are not a whole lot of stats, and you don't have a lot of critical values to call. So here's some examples of critical values that you might call. You would call positive blood cultures, positive spinal fluid gram stains or cultures, a positive Cryptococcus antigen test or culture, positive malaria, group A strep, which is Streptococcus pyogenes from a sterile site, any positive acid-fast smears that would indicate Mycobacterium is present, Streptococcus agalactiae, which is group B strep or herpes virus from a genital site, and detection of significant pathogens. Now, for significant pathogens, there will be a list, a poster, something probably on the wall in the microbiology lab. So if you're not sure, you can go and look at that list to see if it's a significant pathogen, and does it need to be reported to the health department, and then the health department will report it to the state health department, and the state health department will report it to the Center for Disease Control. So there is a chain of command and a chain of information set up for those. So very, very important. So I want to go back and make sure that you know that the first quadrant is the one where we expect the most concentration of bacteria, and the fourth quadrant is the one where we expect the most isolated colonies. Very important. And what techniques do we use to isolate bacteria? The isolation or quadrant streak is used to isolate bacteria, and the quantitation streak is used to quantitate or do a colony count. All right. Well, I enjoyed this chapter, and I hope you did too, and I am ready to really get into it. I love microbiology. That is the end of Chapter 6, Part 2. I forgot to add one thing on rejected specimens. So what do you do if a physician just really insists that you go ahead and do a culture? Even if you try to reject it and you explain to him this has been sitting out for 3 days or 2 days, or this wasn't labeled, and he just throws a fit, you can go ahead and plate it and set it up and add a comment explaining that there would be a potential contaminant. So, you just have to kind of go with the flow and be professional and take into consideration what the specimen is. Was it one of those hard to collect samples or not? So this is what is going to separate us from the machines, and we will always be a valuable part of the healthcare team in the laboratory. All right, now we're finished.